The year is 1888. A young anatomist, Santiago Ramon y Cajal, publishes Estructura del Sistema Nervioso de los Hombres y los Vertebrados, a monumental contribution which did, in fact, structure our understanding of the nervous system for generations to come. This essay undertakes a comprehensive evaluation of the work's claims, meticulously examining its arguments, evidence, and methodology. We will endeavour to expose the intricate architecture of Cajal's reasoning, acknowledging both its triumphs and the inevitable limitations inherent in any pioneering scientific endeavour. Our journey prioritised direct engagement with the text, minimising mere biographical details or historical context, except where they illuminate the validity of specific claims within them. Section 1: Initial Observations and Staining Techniques. Cajal begins by outlining various methods used to visualize nervous tissue. A central claim emerges immediately: the importance of metallic impregnation techniques, particularly those developed by Camillo Golgi. Cajal acknowledges Golgi's method's ability to stain individual neurons in their entirety, a feat previously unattainable. However, he argues, Golgi's interpretation of the stained images is flawed. Cajal contends that the Golgi method, despite its limitations in consistently staining all cells, reveals discrete neuronal units, rather than a continuous reticulum. This is not merely a technical point, it represents a fundamental disagreement about the organization of the nervous system. Cajal details refinements he implemented in the Golgi method, including variations in silver nitrate concentration and fixation protocols, all aimed at enhancing the clarity and reliability of cellular staining. He claims these modifications improved the selective staining of neurons, allowing for more detailed observation of their morphology, particularly the fine axonal and dendritic processes. The evidence presented initially rests on comparative images, showcasing neurons stained with standard Golgi techniques versus those treated with Cajal's refined methods. The assessment centers on his claim of improved resolution and clarity of neuronal processes, although the subjective nature of image interpretation at this stage must be acknowledged. Section 2: Morphology of neurons in the spinal cord. Cajal turns his attention to the spinal cord, a region, he argues, provides a relatively simple, accessible model for understanding neural organization. He describes various types of neurons present in the spinal grey matter, classifying them based on their size, shape, and location. A key claim in this section lies in his characterization of motor neurons, large cells situated in the ventral horn, possessing long axons that project to muscles. He asserts a direct connection between these motor neurons and muscle fibers, mediating voluntary movement. He also describes the existence of interneurons, smaller cells that connect sensory and motor neurons within the spinal cord. He argues these interneurons play a crucial role in integrating sensory information and coordinating motor responses. His descriptions are supported by drawings of neurons observed under the microscope, detailing the characteristic branching patterns of their dendrites and the course of their axons. He meticulously traces the pathways of these processes, providing evidence for the connections between different neuronal types. The assessment hinges on the accuracy of his representations and the validity of his interpretation of neuronal connectivity based on microscopic observations. The difficulty lies in the limitations of the Golgi stain, which only reveals a fraction of neurons at any given time, making it challenging to determine the precise nature of the connections between them. He acknowledges the potential for misinterpretation, but argues the consistency of his observations across multiple preparations lends credence to his conclusions. A subtle implication of his depiction of distinct neuronal types, characterized by specific morphologies and connection, a rejection of the prevalent reticular. Section 3: Organization of the cerebral cortex. The cerebral cortex, being the most complex structure in the nervous system, presents a significant challenge to Cajal. He divides the cortex into distinct layers, each containing a characteristic population of neurons. He describes pyramidal cells, the most prominent neuronal type in the cortex, arranged in a columnar fashion. He claims the apical dendrites of these pyramidal cells extend towards the cortical surface, while their axons project downwards, connecting with other cortical areas or subcortical structures. He posits that these pyramidal cells are the primary processing units of the cortex, responsible for higher-order cognitive functions. He also describes other types of neurons present in the cortex, including granule cells, stellate cells, and horizontal cells of Cajal. He argues each of these cell types contributes to the complex circuitry of the cortex. His observations are again based on Golgi-stained preparations, meticulously drawn to illustrate the intricate branching patterns of neural processes. He attempts to establish connections between different cortical layers, tracing the paths of axons and dendrites as they traverse the cortical columns. The assessment requires careful consideration of the limitations of his methodology. The Golgi stain, while invaluable, only reveals a sparse representation of cortical neurons, making it difficult to fully understand the complex interconnections between them. Cajal acknowledges this limitation, stating that his description of cortical circuitry is necessarily incomplete. However, he asserts the fundamental principles of neuronal organization he identifies provide a solid foundation for future risks. Section 4: Cerebellar structure and function. The cerebellum, with its highly ordered structure, offered a more tractable subject for study than the cerebral cortex. Cajal describes the characteristic arrangement of neurons in the cerebellar cortex, including Purkinje cells, granule cells, and Barcyt cells. He claims the Purkinje cells, large neurons with elaborate dendritic trees extending into the molecular layer, are the principal outward neurons of the cerebellar cortex. He asserts their axons project to the deep cerebellar nuclei, which in turn send projections to other parts of the brain. He argues the granule cells, small neurons located in the granular layer, receive input from mossy fibers and project to Purkinje cells via parallel fibers. He posits the Barcyt cells, interneurons located in the molecular layer, inhibit Purkinje cells, providing a mechanism for regulating their activity. He carefully details the synaptic contacts between these different cell types, based on his observations of Golgi-stained preparations. He describes the parallel fibers making synaptic contact with the dendritic spines of Purkinje cells, and the Barcyt cell axons forming Barcyt-like structures around the cell bodies of Purkinje cells. The assessment of this section relies on the validity of his interpretations of synaptic connections based on microscopic observations. The Golgi stain does not directly visualize synapses, it only reveals the morphology of neurons and their processes. Cajal infers the presence of synapses based on the proximity of neuronal processes and the patterns of dendritic spines. He argues the highly ordered structure of the cerebellar cortex makes it easier to identify these synaptic connections with greater certainty than in other brain regions. Still, the inherent limitations of inferring synaptic connectivity from static images must be acknowledged. Section 5: Retina and visual pathways. Cajal extends his analysis to the retina and visual pathways, seeking to understand the neural basis of vision. He describes the various cell types present in the retina, including photoreceptors, bipolar cells, ganglion cells, and amacrine cells. He claims the photoreceptors, located in the outermost layer of the retina, are responsible for detecting light. He asserts the bipolar cells transmit signals from photoreceptors to ganglion cells. He argues the ganglion cells, whose axons form the optic nerve, transmit visual information to the brain. He painstakingly traces the pathways of axons originating from retinal ganglion cells, demonstrating their projection to the lateral geniculate nucleus of the thalamus, and from there to the visual cortex. He outlines the laminar organization of the retina, detailing the connections between different cell types within each layer. He posits that the retina is not simply a passive receiver of light, but an active processor of visual information. Here, the assessment depends on the accuracy of his tracing of axonal pathways. The Golgi stain allows him to visualize individual axons, but it does not provide information about the direction of signal transmission. He infers the direction of signal transmission based on the morphology of the neurons and the location of their synapses. He hypothesizes that visual information flows in a specific direction through the retina, from photoreceptors to bipolar cells to ganglion cells. This hypothesis is not directly proven by his observations, but it is consistent with the anatomical evidence he presents. Section 6 Olfactory system, a unique case. Cajal dedicates considerable attention to the olfactory system, recognizing its distinctive organization. He describes the olfactory bulb, the first processing station for olfactory information, and identifies its key neuronal components: mitral cells, tufted cells, and granule cells. He claims mitral cells, the primary output neurons of the olfactory bulb, receive input from olfactory receptor neurons. In the nasal epithelium, he asserts the tufted cells, smaller neurons located in the outer layers of the olfactory bulb, also receive input from olfactory receptor neurons, but their axons project to different brain regions compared to mitral cells. He argues the granule cells, small densely packed into neurons, modulate the activity of mitral cells, providing a mechanism for sharpening olfactory discrimination. He observes that the olfactory receptor neurons, unlike other sensory neurons, project directly to the brain, bypassing the thalamus. He posits that this unique anatomical arrangement reflects the ancient evolutionary origins of the olfactory system. The assessment rests on the validity of his claims regarding the unique anatomical features of the olfactory system, particularly the direct projection of olfactory receptor neurons to the olfactory bulb. Cajal supports this claim with careful tracing of axonal pathways, demonstrating the absence of a thalamic relay station in the olfactory pathway. This discovery challenges the then prevalent view that all sensory information must pass through the thalamus before reaching the cortex. Section 7 Neurogenesis and Neuronal Plasticity. While the bulk of Cajal's work focuses on the established structure of the nervous system, he also cautiously ventures into the realm of neurogenesis and neuronal plasticity. He observes the presence of immature neurons in the developing nervous system, suggesting that new neurons can be added throughout life. He claims these immature neurons migrate to their final destinations and differentiate into mature neurons. While the evidence for adult neurogenesis at this time was limited, Cajal's intuition here points towards a concept only fully appreciated a century later. He also observes changes in neuronal morphology in response to experience, suggesting that the nervous system is not static, but capable of adapting to changing environmental demands. He acknowledges that the extent of neuronal plasticity is limited in the adult brain, but he argues that it plays a crucial role in learning and memory. The assessment of these claims regarding neurogenesis and plasticity requires a nuanced perspective. Cajal's observations were limited by the available technology, and he could not definitively prove that new neurons are generated in the adult brain or that neuronal morphology changes in response to experience. However, his observations laid the groundwork for future research in these areas, which has since confirmed the existence of both adult neurogenesis and neuronal plasticity, albeit to varying degrees in different brain regions. Section 8 A Defense of Neuron Doctrine. Throughout his work, Cajal forcefully defends the neuron doctrine, the idea that the nervous system is composed of discrete, individual cells. This doctrine was in direct opposition to the reticular theory championed by Golgi and others, which held that the nervous system is a continuous network of interconnected fibers. Cajal systematically refutes the arguments of the reticularists, pointing to the clear evidence of distinct neural cell bodies, axons, and dendrites revealed by the Golgi stain. He argues that the Golgi stain, while not perfect, provides the strongest evidence against the reticular theory. He acknowledges that the Golgi stain only reveals a small fraction of neurons, but he argues that the consistency of his observations across multiple preparations demonstrates the universality of the neuronal structure. He argues that the reticular theory cannot explain the complexity and specificity of neuronal connections observed in the nervous system. Further, Cajal was convinced that the directionality of neural transmission was inconsistent with the reticular theory. A continuous reticulum, he argued, would not support the precise, directed flow of information that he proposed. This section effectively summarizes the central argument that underpins his entire work, that the nervous system is not a seamless web, but a mosaic of individual, interacting units. The assessment requires recognizing the context of scientific debate at the time. The reticular theory was a formidable opponent, supported by prominent scientists. Cajal's unwavering defense of the neuron doctrine, based on meticulous observation and careful reasoning, ultimately prevailed, transforming our understanding of the nervous system. Section 9: Comparative Anatomy and Evolutionary Insights. Cajal broadens his scope to encompass comparative anatomy, examining the nervous systems of various vertebrate species. He claims that the fundamental principles of neuronal organization are conserved across species, suggesting a common evolutionary origin. He observes similar neuronal types and connection patterns in the brains of different vertebrates, from fish to mammals. He argues that the complexity of the nervous system increases with increasing phylogenetic complexity, reflecting the evolution of more sophisticated cognitive abilities. He observes that while the basic architecture remains the same, the size, number, and complexity of neurons increase in more advanced species. He posits that the evolution of the nervous system has involved the elaboration and refinement of existing neuronal circuits, rather than the creation of entirely new structures. The assessment requires considering the limitations of comparative anatomy as a method for understanding evolutionary relationships. Cajal's observations are based solely on anatomical data, and he does not have access to the genetic or molecular tools that are now used to study evolution. However, his observations provide valuable insights into the evolution of the nervous system, suggesting that the basic building blocks of the nervous system have been conserved throughout vertebrate evolution. Section 10: Methodological Limitations and Future Directions. Cajal acknowledges the methodological limitations inherent in his work. He emphasizes the limitations of the Golgi stain, noting that it only reveals a small fraction of neurons and does not provide information about the direction of signal transmission or the nature of synaptic connections. He calls for the development of new techniques that can overcome these limitations and provide a more complete understanding of neuronal structure and function. He encourages future researchers to investigate the functional properties of individual neurons to determine how they encode and process information. He suggests using electrophysiological methods to measure the electrical activity of neurons and to study the effects of different stimuli on their activity. He acknowledges that his work is only a starting point and that much remains to be discovered about the nervous system. This section is particularly significant because it shows Cajal's intellectual honesty in his commitment to scientific progress. He does not overstate the significance of his findings, but rather acknowledges their limitations and points the way for future research. Conclusion: structure of the nervous system of man and the vertebrates constitutes a foundational text in the history of neuroscience. Cajal's meticulous observations, supported by his artistic renderings, provided compelling evidence for the neuron doctrine. While his work was limited by available technology, his insights into neural organization, synaptic connectivity, and neurogenesis laid the groundwork for subsequent research. The essays section by section evaluation confirms the enduring value of Cajal's claims, even as it acknowledges the inevitable complexities and unanswered questions that remain in the quest to understand the most intricate structure in the known universe. Cajal's profound contribution lies not only in his discovery of the neuron as the fundamental unit of the nervous system, but also in his unwavering commitment to rigorous observation, careful reasoning, and intellectual honesty, qualities that continue to inspire neuroscientists today. His work, therefore, stands as a testament to the power of meticulous anatomical investigation, a legacy that continues to shape our understanding of the brain and its remarkable capabilities.